I want you to picture something that is, well, it's incredibly familiar to all of us. You know, you're sitting in a dimly lit room, right? Surrounded by friends and family.
Yeah, and they're all singing that exact same song we all know.
Exactly. And right in front of you is a cake. It's glowing with the light of a very specific number of candles. So you take a deep breath, you close your eyes, you make a wish, and you blow them out.
It's a celebration. I mean, we are totally conditioned to view it as this triumph, right? A triumph of surviving another trip around the sun.
Right. But I want you to consider the literal biological reality of your body at that exact moment. Because every single time you celebrate a birthday and, you know, blow out a candle, you are actually marking another year of profound, measurable changes happening at the microscopic level.
Oh, absolutely. Across all your organ systems.
Yeah. Your cells are losing their baseline ability to repair themselves. With every passing year, your DNA is accumulating this structural damage that its built-in repair mechanisms just can't fully fix anymore.
You're experiencing a progressive degradation of cellular identity.
Yeah.
I mean, it's essentially a biological countdown clock ticking away in the background while you eat your slice of cake.
Which is pretty grim when you put it like that.
It forces a completely different perspective on the whole birthday ritual, doesn't it? Because for the entirety of human history, that degradation has been universally accepted as a fundamental law of nature.
Right, the whole circle of life thing.
Exactly. You're born, you reach reproductive maturity, you age, and then, well, your physiological systems eventually fail. It's always been considered as inevitable as thermodynamics.
The idea that complex systems just naturally trend toward entropy over time.
Precisely. Like a car rusting in a field.
But, and this is why we're here today, we have to challenge that assumption right here at the start. What if aging isn't a fundamental law of nature at all?
Right.
What if it's not just passive entropy, but actually an active biological process, like a flaw in our biological operating system that we are finally, right now, learning how to isolate and, you know, potentially modify?
That's the billion-dollar question.
Welcome to this deep dive into the science of longevity. Today, we are examining this massive collision between cutting-edge biological research and an enormous multi-billion-dollar industry.
Yeah, we're going to look at the latest laboratory breakthroughs, the clinical trial data, and, honestly, the massive commercial hype to figure out what the science actually says about human health span.
To do that effectively, though, we really have to establish a crucial foundation about how we conceptualize the aging process itself.
Right, because the dominant paradigm for most of modern history has been that wear and tear model I mentioned earlier.
A car rusting in the field.
Exactly. The assumption was that your body is essentially like a machine. You operate a machine for long enough, the parts experience friction, they degrade, the materials fatigue, and eventually the system just breaks down simply because it's been used.
It's just a passive consequence of existing in the physical world.
Yeah, but the biological consensus is shifting violently away from that model right now.
Violent is a good word for it. It's a total paradigm shift.
Because aging is not simply passive wear and tear. It's not just structural fatigue. Aging is an active biological process. It's driven by very specific, measurable, and highly orchestrated cellular mechanisms.
And that distinction, that is everything. If aging is merely random, chaotic decay governed by the laws of physics, well, intervention is effectively impossible. You can't stop entropy.
You really can't.
But if aging is an active process, like if it's a biological program that executes based on specific genetic instructions, that means the mechanisms can be understood.
And if they can be understood, they are potentially programmable.
Exactly. Everything we discuss in this deep dive has to be viewed through this exact lens. Aging as a modifiable program. If we can map the molecular pathways of that program, the underlying assumption of modern longevity science is that we can eventually intervene in them.
So let's examine the actual architecture of that program, right?
Yeah.
What are the physiological realities of aging if we completely toss out the wear and tear model?
The research kind of coalesces around this framework that's often called the hallmarks of aging.
Right. These are the primary interrelated processes that drive the progressive loss of your physiological integrity. And we really need to explore these mechanisms beyond just the superficial level.
Starting with, I guess, the baseline instability of the genome itself, the DNA damage.
Yeah, the accumulation of DNA damage. And look, we're not just talking about external trauma here.
Like getting a sunburn.
Right. Environmental factors like UV radiation or chemical exposure are obvious culprits. But the more insidious damage comes from the totally normal metabolic processes of simply being alive.
Just breathing and eating.
Exactly. Cellular respiration produces reactive oxygen species. These are byproducts that actively interact with and damage your DNA structure.
Plus, you have the fundamental replication problem, right? Every time a cell divides, it has to copy billions of base pairs of DNA.
Billions. And the replication machinery is astonishingly accurate, but it is not perfect.
So over decades of constant division, you get errors.
You get base pair mismatches, you get double strand breaks. Now, the cell does have robust repair pathways to deal with this.
But as we age, those repair mechanisms get sloppy.
They decline in efficiency, yes. The structural integrity of the genome degrades. The code itself accumulates errors. And this leads directly to a really highly publicized aspect of this damage, the attrition of telomeres.
Oh, telomeres. You hear about these all the time in longevity podcasts and articles. They're usually described with that shoelace analogy, right?
The plastic caps at the end of your shoelaces, yeah. It's a simplification, but the actual mechanics are fascinating. Telomeres are these repetitive sequences at the ends of our linear chromosomes.
Because the replication machinery can't copy the very end of the DNA strand.
Exactly. It's called the end replication problem. So with each successive cellular division, a tiny little portion of that protective telomere sequence is lost.
It essentially functions as a countdown clock for the cell.
A mitotic clock, precisely. Once the telomere gets too short, the chromosome is exposed. And the cell's internal monitoring systems, they don't see this as a normal structural feature. They see it as severe DNA damage.
Like a massive alarm goes off, the cell panics.
It totally panics. It interprets its own natural replication cycle as a catastrophic break. And this is where the evolutionary programming kicks in hard.
Because if that cell kept dividing with unprotected, unstable chromosomes,
it would become cancer. The genomic chaos would almost certainly lead to malignant transformation. So the biological program dictates a failsafe.
It hits the emergency brake.
Yes, it triggers a permanent arrest of the cell cycle. And that permanent arrest is what we call cellular senescence.
Cellular senescence. Okay, so instead of just dying and getting cleared out, the cell just stops.
Right. It doesn't die through the normal programmed cell death pathways. Instead, it enters this weird state of suspended animation. It's still metabolically active, but it can never divide again.
So in the short term, that's great. It stops a damaged cell from turning into a tumor.
In the context of evolutionary biology, it is a highly successful tumor suppression mechanism.
But the research indicates that this evolutionary failsafe becomes a massive liability as we get older, right? Because these senescent cells, they're constantly referred to in the literature as zombie cells.
Zombie cells is a very apt term. They refuse to die, and they completely stop doing their specialized jobs.
So a senescent skin cell isn't making collagen anymore.
No. And a senescent cell in your blood vessels isn't keeping the arteries elastic anymore. But the loss of function is really only half the problem.
It's what they start doing instead that's so bad.
Yes. The destructive aspect is what the cell actively begins to secrete into the body. Senescent cells develop what researchers call a senescence-associated secretory phenotype.
SASP.
SASP. They basically start pumping a complex cocktail of pro-inflammatory cytokines, chemokines, and tissue remodeling enzymes into the surrounding area.
So they're actively degrading the healthy tissue around them.
They are. And the truly insidious part is that this inflammatory signaling can actually induce senescence in neighboring perfectly healthy cells.
Wow. So the zombie cells infect the healthy cells. It's like a localized biological wildfire.
That's a great way to put it. An evolutionary mechanism designed to suppress tumors when you're young ends up creating a chronic inflammatory burden that destroys your tissues when you're old.
Okay, so we have DNA damage, telomere shortening, and these zombie cells, but we also have to talk about the epigenome, right?
Absolutely. The epigenome dictates how your genetic code is actually utilized.
Because the DNA is just the blueprint, but the epigenome is the construction manager reading the blueprint.
Exactly. It's the chemical modifications that regulate gene expression. It determines which genes are turned on and which are silenced. I mean, a neuron in your brain and a hepatocyte in your liver, they have the exact same DNA.
But they look and act completely differently because of their epigenetic profiles.
Right. But as we age, that epigenetic regulation becomes fundamentally dysregulated. We see global changes in these patterns.
Areas of the genome that should be tightly packed away and silenced suddenly open up and become active.
And essential regions that should be active get shut down. The consequence is this profound loss of cellular identity. A cell literally forgets its highly specialized function and starts expressing the wrong proteins.
Which is known as epigenetic drift.
Yes. And when you synthesize all these interconnected mechanisms, the genomic instability, the telomeres, the senescence, the epigenetic drift, you observe a systemic cascading failure of information maintenance.
The biological program is essentially losing its structural integrity. It can't read its own code correctly anymore.
And understanding this complex interplay, it forces us to confront a vital distinction in this field, the critical difference between lifespan and health span.
This is such an important concept because lifespan is just chronological, right? It's just the absolute duration of how long you exist. If medical intervention keeps your heart beating until you are 95, your lifespan is 95 years.
But health span is the proportion of that lifespan spent in optimal health, free from chronic diseases and the severe functional decline that we associate with aging.
And modern medicine over the last century has been incredibly good at extending lifespan. We have antibiotics, complex surgeries, chronic disease management that stops you from dying acutely.
But we have been largely ineffective at extending health span at the same rate.
So the consequence is we get this prolonged period of morbidity at the end of life. People are spending 20-30 years managing cascading metabolic dysfunction, heart disease, neurodegeneration, frailty.
They are surviving, yes, but their physiological independence is severely compromised.
So the primary objective of longevity research isn't just to tack on more years at the end, it's the compression of morbidity.
The goal is to intervene in the biological program so that health span closely aligns with lifespan, to keep you vigorous and functional until right before the very end.
Which brings us to the comparative biology, and this is where I find the whole aging as a program premise undeniable. Because when we look at the natural world, we see that evolution has already engineered totally different settings for this program.
Humans aging at this specific rate is not a universal constant.
No, the examples are crazy. You have certain species that exhibit negligible senescence, like those deep-water sharks that live for centuries with barely any decline in function.
And the naked mole rat is a particularly fascinating model organism for this.
They are so weird, but their resistance to aging is staggering. They live like 10 times longer than other rodents their size.
And the research points to highly specific mechanistic adaptations, like their cells exhibit extreme contact inhibition.
Which makes them uniquely resistant to spontaneous tumor formation.
Right. They have evolved superior mechanisms to maintain the stability of their proteins, highly efficient DNA repair. They literally demonstrate that the biological machinery required for extreme longevity already exists in nature.
It's just a matter of evolutionary programming, not some violation of physics. And I find that unbelievably empowering.
It's a very optimistic way to view it.
But think about it. If aging is governed by specific cellular pathways, and we have physical proof in these animals that those pathways can be optimized for vastly longer periods, then human aging is essentially just an engineering problem.
In theory, yes.
If the program is generating epigenetic noise and accumulating senescent cells, we theoretically just need to develop the biochemical tools to clear the noise and patch the code.
The theoretical framework is sound. The biological principles do support the possibility of intervention.
So it's not like just updating your phone's software.
Not at all. You cannot isolate one pathway without triggering a cascade of secondary effects. Take the telomere issue we discussed. If telomere attrition limits cellular division, a logical intervention might be to just upregulate telomerase.
Right, the enzyme that rebuilds telomeres. If the clock is running out, just rewind the clock. Force the cells to keep dividing indefinitely.
But the biological roadblock to that is massive. As we just established, cellular senescence evolved specifically as an anti-cancer mechanism.
Right.
If you artificially upregulate telomerase in a cell that has already accumulated significant DNA damage, you completely remove the emergency brake. You allow a mutated cell to divide uncontrollably.
So you'd solve the cellular aging problem, but immediately trigger lethal malignancies everywhere.
The oncological risk of systemic telomerase activation is absolutely prohibitive. It's a biological double-edged sword. It's the concept of antagonistic pleiotropy.
Antagonistic pleiotropy. What does that mean in this context?
It means that genes or processes that give you a survival advantage early in life, like a robust cell arrest mechanism to stop cancer while you're young enough to reproduce, they become actively detrimental late in life when those senescent cells pile up.
Oh, wow. Because evolution really only optimizes for reproductive success.
Exactly. It doesn't optimize for optimal health at advanced ages. Once an organism has passed its reproductive window and ensured the survival of its offspring, the evolutionary selective pressure to maintain that organism's cellular integrity drops precipitously.
We are fighting against an evolutionary architecture that simply doesn't care what happens to us after we turn 50.
Overcoming millions of years of evolutionary prioritization requires a level of biochemical precision that we are really only just beginning to conceptualize.
Okay, so if aging is this highly complex, interrelated program that evolution actively resists modifying for the long term, we have to look at the researchers who are attempting to force those modifications anyway.
Let's transition to the real science. Let's examine what is actually demonstrating efficacy at the cellular level right now.
The most disruptive area of research right now has to be the field of epigenetic reprogramming.
Without a doubt. We established earlier that aging involves profound epigenetic dysregulation, the noise that obscures the cell's identity. Reprogramming targets that specific mechanism.
The concept here is radical. It's the idea that you can take a fully differentiated, chronologically old cell, like a skin cell, and expose it to a specific sequence of transcription factors that actively remodel its structure.
These factors penetrate the nucleus, and they effectively erase the epigenetic marks. They strip away the accumulated patterns, and by doing so, they force that mature cell to revert all the way back to a state of pluripotency.
Pluripotency meaning it becomes basically identical to an embryonic stem cell.
Yes, capable of turning into any tissue type.
The biological implication of that is just staggering. It proves that the actual genetic code, the hard drive, is largely perfectly intact, even in really old cells. The age isn't hardwired into the DNA sequence, it's just the software running on top of it.
It's a profound realization. But from a therapeutic standpoint, forcing cells into complete pluripotency inside a living organism is disastrous.
Oh, absolutely disastrous.
If you strip a cell of its functional identity while it's still in your liver, the structural integrity of that litter collapses. Plus, highly proliferative pluripotent cells form complex, highly disorganized tumors called teratomas.
Teratomas, yikes. So full reprogramming is a great lab tool, but lethal as a therapy.
Right, which is where the concept of partial reprogramming completely changed the trajectory of the entire field.
The hypothesis behind partial reprogramming is so elegant. Researchers realize that the erasure of these epigenetic marks happens in phases, right?
Yes.
So if you expose the cell to the reprogram factors for just a transient period, like a short controlled pulse instead of continuous exposure, can you clear the accumulated noise without crossing the threshold of losing the cell's identity?
You want to rewind the clock back to a youthful state, but you stop the process just before the cell forgets it's supposed to be a kidney cell.
Exactly. And certain studies in animal models suggest that this partial reversal is actually possible. We really have to talk about the 2025 mouse data here.
It represents a very significant benchmark, yes. Researchers applied a pulsed reprogramming protocol to chronologically old mice.
They used viral vectors to deliver the factors, turning them on intermittently, and they tracked the outcomes over 7 months, which is a massive portion of a mouse's lifespan.
The duration is critical. Transient interventions often fail to produce durable systemic changes.
But the readouts were incredible. They analyzed the tissues, they measured the RNA being expressed, and they found that the gene expression patterns in the liver and kidney tissues of the treated older mice looked just like significantly younger control mice.
The cellular morphology, the actual structural health of the tissue, demonstrably improved.
They weren't just slowing the decline. The data indicates they were actively reversing the biological age of the tissue. It's incredible.
The transcriptomic data and the morphological improvements in those specific murine models are undeniably compelling. The mechanism demonstrates real in vivo efficacy. But I have to ask, has this been proven in humans?
That is the necessary friction here.
It is.
It is so easy to look at reversed biological clocks in a mouse kidney and just immediately assume human immortality is a decade away.
And we must be hypervigilant about that translational gap. Because a mouse is an organism with a fundamentally different metabolic rate, a wildly different lifespan, and a completely different oncological profile than a human being.
The history of medicine is a graveyard of things that cured mice.
Defined by interventions that produce miraculous results in rodents and either failed completely in humans or worse, produce unacceptable toxicity. The scale is vastly different. A transient pulse that rejuvenates a mouse organ over 7 months could theoretically trigger delayed oncogenesis in a human over 5 years.
Because we are manipulating the fundamental regulators of cellular growth.
The margin for error between rejuvenation and malignant transformation is terrifyingly narrow.
Which is why the clinical application of this tech requires so much caution. However, it is a fact that some of the very first human trials utilizing forms of epigenetic reprogramming are underway right now in 2026.
But the parameters of those trials are highly restricted.
Yeah.
They are absolutely not attempting systemic whole-body rejuvenation.
Right. They are highly localized. Like, some trials are targeting the cells in the optic nerve to try and restore vision loss to neuropathies.
Because the eye is an immunoprivileged site, it's relatively isolated from the systemic circulation, which makes it a much safer proving ground for this kind of manipulation. It's a necessary, highly conservative first step.
Now, while reprogramming focuses on restoring cell function, the other major avenue takes a much more aggressive approach, Senolytics.
Ah, yes. Addressing the zombie cells.
If reprogramming is fixing the software, Senolytics are about excising the hardware that is actively damaging the system.
The underlying strategy is selectively inducing apoptosis-programmed cell death in the senescent cells without harming the healthy tissue around them.
And to do that, they have to exploit a specific biological vulnerability, because zombie cells are incredibly resistant to dying naturally.
They are. They upregulate specific survival pathways, which allows them to survive despite carrying severe damage and being in a state of high metabolic stress.
So senolytic compounds are designed to target and inhibit those specific survival pathways.
When you block the anti-apoptotic signals in a senescent cell, the accumulated damage finally triggers the cell to undergo apoptosis.
It essentially forces the zombie cell to complete the death sequence it's been resisting for months.
And the hypothesis is that by clearing out a significant percentage of these cells, you dramatically reduce that SASP, the systemic inflammatory burden. You remove the local toxicity, and you allow the surrounding healthy cells to proliferate and repair the tissue.
And the preclinical data for Senolytics has been robust for years. They've shown improvements in cardiovascular function, metabolism, even cognitive metrics in various animal models.
But the sheer volume of chemical compounds that require testing to find safe and highly selective senolytics is a monumental logistical challenge.
Which is where the intersection of biology and AI is completely altering the timeline. Artificial intelligence in drug discovery is accelerating this exponentially. We aren't just testing thousands of random compounds in petri dishes over 5 years anymore.
We are using in silico predictive modeling. The algorithms analyze massive data sets of molecular structures, predict how they will interact with specific biological targets, and synthesize novel compounds optimized for selectivity.
The 2025 research is a perfect example. They used AI to sift through vast chemical libraries and found multiple novel senolytic candidates. They tested the best ones in vivo on model organisms, and the report indicates that 70% of those AI-selected compounds successfully extended lifespan and improved Healthspan metrics in nematode worms.
That is remarkable.
A 70% hit rate for a predictive algorithm. It's a staggering acceleration of the discovery pipeline. We're moving so fast.
The computational efficiency is undeniably transformative, yes. The AI can identify novel molecular scaffolds that human researchers might never conceptualize. But again, I must interject. Has this been proven in humans?
Okay, fair. Nematode worms are microscopic. They have exactly 959 somatic cells.
Precisely. Extending the lifespan of C. elegans is a valuable demonstration of biological principle, but it is light years away from resolving the complexities of human pharmacology.
The AI doesn't know what a human liver looks like in 50 years.
The AI can predict a binding affinity perfectly, but it cannot always predict off-target toxicity in a complex mammalian system, or how the compound will interact with the human microbiome, or how it alters immune responses over a decade. We must unequivocally state, no supplement, therapy, or intervention, whether discovered by AI or human ingenuity, is currently proven to reverse aging in humans.
That is the stark reality. The scientific method demands decades of patience. It demands longitudinal studies, enormous sample sizes, and a terrifyingly high tolerance for failure.
The biology is advancing, the computational tools are unprecedented, but the timeline for definitive human proof is inherently slow.
Which brings us to a massive, massive structural conflict, because the scientific method requires decades of patience, but the commercial market demands immediate capitalization.
Let us pivot to examine the industry that has materialized around these biological concepts.
Yes, let's talk about the hype machine. The disconnect between the methodical pace of the laboratory and the absolute frenzy of the consumer market is staggering. We are looking at a global anti-aging market that generated over $85 billion in 2025.
And projections are pushing it toward $120 billion by the end of the decade. That volume of capital creates a powerful gravitational pull.
It totally incentivizes the premature commercialization of preliminary data.
When there is $85 billion actively seeking a return on investment, the nuance and the conditional language of the actual science are frequently discarded in favor of aggressive marketing.
So what is that $85 billion actually buying? Because if, as we just firmly established, there are 0 proven age reversal therapies for humans, what are all these people putting in their bodies?
The market is currently dominated by a proliferation of dietary supplements, complex biohacking regimens, the off-label use of existing pharmaceuticals, and biological age testing kits.
And the vast majority of these interventions lack the robust, randomized, placebo-controlled human trials required to definitively prove longevity benefits.
Take the supplement market as a prime example. Proponents suggest that specific coenzyme precursors molecules designed to theoretically boost mitochondrial function can mimic the effects of caloric restriction and extend health span.
Right. And the mechanistic rationale is based on legitimate biology. Optimizing mitochondrial efficiency is a valid target.
But proponents frequently extrapolate in vitro cellular data or short-term rodent studies into definitive claims of human age reversal. They market these compounds with language co-opted directly from the rigorous science we discussed.
They use terms like epigenetic optimization and cellular rejuvenation on a bottle of pills you can buy at a grocery store.
It is a sophisticated linguistic sleight of hand. They blur the crucial distinction between interventions that might subtly support general metabolic health, which is effectively health span extension, and interventions that actively reverse the biological program of aging.
And the structural economics of the longevity startup ecosystem practically mandate this overpromising, don't they?
They really do. Conducting the kind of longitudinal human trial required to prove an intervention extends health span by a decade would require hundreds of millions of dollars and decades of observation.
But the primary mechanism for funding early-stage biotech is venture capital.
Exactly. And venture capital operates on a rigid timeline. Funds typically require an exit or a significant return on investment within a 7 to 10-year window.
So there's a fundamental mismatch between the biological timeline required to prove it works and the financial timeline required to get the money to study it.
To attract that necessary VC funding, startups are under immense pressure to present highly aggressive timelines. They highlight a successful intervention in a mouse, they point to AI drug discovery, and they suggest to investors that commercial application is imminent.
They are forced to sell the absolute best-case, highly speculative scenario as a near certainty.
And that aggressive narrative inevitably trickles down to the consumer market. It fuels the biohacking extreme.
Oh, the biohacking subculture is absolutely fascinating to me. You have individuals structuring their entire existence around theoretical longevity optimization. They're consuming dozens of unproven compounds every single morning,
Based on the thinnest of preliminary data.
They're subjecting themselves to extreme temperature variations, cold plunges, saunas. They're monitoring continuous glucose levels despite having no metabolic dysfunction whatsoever.
They're measuring dozens of unvalidated biomarkers constantly. It is an attempt to exert absolute control over a biological program that we only partially understand.
But the irony is profound. They're enduring significant daily psychological and physical stress, which itself is a known accelerator of cellular aging, based on the mere guess that these combined interventions might theoretically extend their lifespan.
They are actively sacrificing their current quality of life for the highly speculative promise of a longer future.
I mean, I'm taking 50 pills, sitting in an ice bath, tracking my blood sugar, and I'm miserable, but maybe I'll live to 110.
We must challenge this hype vigorously. The underlying science of cellular senescence and epigenetic regulation is genuinely revolutionary. But the commercial translation of that science is currently rife with extrapolation.
Right. Selling an unvalidated epigenetic age test to a consumer and implying that a proprietary supplement blend will reverse their clock based on a single observational study is fundamentally exploitative.
We have to maintain a state of dual awareness. We can be genuinely awestruck by the potential of partial reprogramming, while simultaneously being intensely critical of companies selling over-the-counter pills that claim to leverage that biology today.
It requires a really highly sophisticated level of skepticism from the public.
It's a difficult environment to navigate. However, for the sake of intellectual exploration, let's assume that the rigorous science eventually bridges the translational gap.
Let's strip away the venture capital hype and the premature supplements.
Right. Suppose that in 20, 30, or 50 years, we successfully develop highly targeted Senolytics, and safe epigenetic reprogramming protocols that definitively and dramatically extend human health span.
Suppose the science fully delivers on its ultimate promise.
If we fundamentally alter the biological constraints of the human species, we instantly destabilize the societal, economic, and ethical structures that have been built entirely around a roughly 80-year lifespan.
Which introduces questions that are arguably way more complex than the molecular biology itself.
Absolutely.
We have to engage with the hard questions, starting with the most immediate systemic crisis: access, and the potential for a biological underclass. If these interventions become a reality, the initial development and manufacturing costs will be astronomical. We are talking about highly personalized, complex, genetic and cellular therapies.
They will not be mass-market generic interventions upon initial approval. The pricing structure will inherently restrict access to the ultra-wealthy.
And throughout human history, inequality has manifested in resource distribution, political power, quality of life. But mortality has always been the ultimate equalizer.
Death comes for everyone.
Exactly. The billionaire and the impoverished individual both eventually succumb to the same biological degradation. But if we introduce therapies that allow individuals with vast financial resources to live to 140 in vigorous health, while the economically disadvantaged continue to suffer the morbidities of aging and die at 75, we create a literal biological stratification of the human species.
The societal friction generated by that level of disparity would be unprecedented.
Right.
However, one must also consider the economic counterargument regarding the massive cost of aging itself.
What do you mean?
The current medical paradigm spends an overwhelming percentage of global healthcare resources managing the chronic decline of the elderly.
Right. The economic burden of managing late-stage cardiovascular disease, neurodegeneration, systemic frailty, it's catastrophic.
So if a therapeutic intervention successfully compresses morbidity, if it keeps a population biologically young and independent until right before the end of life, the savings to the global healthcare infrastructure could theoretically offset the cost of the therapies over time.
Eventually driving the price down and democratizing access.
Precisely.
And that speaks to the profound moral imperative driving the research in the first place. When you observe the reality of aging, the loss of cognitive function, the physical pain, the immense psychological suffering of families watching loved ones slowly disintegrate, it looks exactly like a devastating disease.
It does.
If we possess the scientific capacity to treat that disease, to preserve the accumulated wisdom and physiological dignity of the individual, isn't there an absolute moral obligation to pursue it, regardless of the initial economic turbulence?
That is a deeply compelling moral position centered on the alleviation of individual suffering. But the ethical analysis must also encompass the macro-level consequences. What happens to the architecture of society if a massive cohort of the population begins routinely living to 150?
The structural implications are terrifying. The entire concept of retirement and intergenerational resource transfer collapses.
Pension systems, social security frameworks, and workforce dynamics are all mathematically calibrated to a specific ratio of working years to dependent years. If lifespan expands dramatically, individuals cannot realistically retire at 65. The economic dependency ratio would crush the working-age population.
So the expectation shifts. You must work until you are 110, but consider the impact on generational mobility and innovation. If the senior executives, the tenured academics, and the political leadership remain vital, entrenched, and active for an entire century, the pathways for advancement for younger generations are completely blocked.
Generational turnover is the primary engine of social and scientific progress. New cohorts challenge entrenched paradigms and introduce novel perspectives.
Right. If the biological turnover ceases, society risks profound ideological and institutional stagnation.
A gerontocracy, even a biologically healthy one, could severely stifle adaptation.
And we can't ignore the sheer arithmetic of population dynamics. If global birth rates remain relatively stable, but the mortality rate plummets because individuals are living twice as long, the compounding pressure on global resources, agricultural output, and environmental stability becomes an existential threat to the species.
Which forces us to confront a fundamental philosophical question regarding the nature of the biological program itself. Is aging purely a pathological process that demands a cure? Or is the constraint of mortality a necessary feature for the vitality of the species as a whole?
It's the argument that boundaries define meaning. Does the urgency to innovate, to create art, to form deep connections, stem from the subconscious recognition that our time is strictly limited? If you have an indefinite horizon, does the imperative to act dissipate?
It's a profound inquiry. A species that does not experience biological turnover may prioritize risk aversion and preservation over exploration and growth. The drive that propels humanity might be intrinsically linked to its biological fragility.
Both of these positions, the desperate moral necessity to alleviate the profound suffering of aging and the terrifying sociological consequences of a stagnant immortal society, they feel completely irresolvable. There is no clear equilibrium. We are rushing toward a biological threshold without the ethical frameworks to manage the transition.
And neither of us possesses the definitive answer to these sociological dilemmas. And that uncertainty is perhaps the most crucial takeaway. The biological capability is accelerating far faster than our societal capacity to integrate it.
Which brings us to the reality of the immediate horizon. If we step back from the philosophical extrapolations of 150-year lifespans, what is the grounded, realistic projection for the listener, based on the rigorous science? What is actually developing in the near term?
If we synthesize the current trajectory of the clinical research, the period between 2026 and 2030 will be a critical inflection point. This is the window where the foundational human trials for localized epigenetic reprogramming and highly selective senolytic compounds will begin to yield substantial data.
This is when we find out if the biology translates.
Precisely. We will begin to discern whether the mechanisms that prove so effective in murine models can be safely titrated for human physiology. But the data will likely be incremental. We will not see an overnight cure for systemic aging.
The most scientifically grounded conclusion we can draw today is that meaningful health span extension is a highly probable outcome of this research. The capability to modulate the biological program, to delay the onset of chronic disease, and to maintain physiological vigor for a much larger percentage of our natural lifespan that is supported by robust mechanisms.
Achieving a high quality of biological function well into our 80s or 90s is a realistic objective for contemporary medicine. However, we must continuously reiterate the conditionality of the extreme claims. Dramatic expansion of maximum human lifespan or the systemic reversal of biological age in a human being remains entirely speculative. The biological hurdles are massive, and the clinical proof simply does not exist yet.
We are fundamentally transitioning how we view our own biology. We are moving away from the passive acceptance of wear and tear and beginning to understand the code of our own degradation.
The potential to modify that code is real, but the timeline requires intense scientific rigor, not venture capital hype.
It demands a rigorous balance, a profound respect for the biological possibilities, coupled with severe skepticism of commercial entities claiming to have solved the problem today.
That is the line we have to walk as this science evolves.
We have explored the genomic instability, the senescence, the reprogramming breakthroughs, the $85 billion hype machine, and the terrifying societal implications. But as we conclude this deep dive, I want to step away from the data and the macroeconomics. I want to leave you, the listener, with a deeply personal question to consider based on everything we have examined today. If you could live to 150 in good health, would you want to? And what would you do differently right now if you knew you could?